Thanks for coming. This is the seventh and last talk in this series dealing with cells. And in some sense, I hope I've properly understood that I was saving the best part to the end. I'll be talking about some recent research results that's going on with cells. And they're really, I think, quite interesting. That figure that I've put there on the talk is the thing that drew me into this whole game some 5 or 6 years ago. It's a very dramatic picture or image of a viruses attacking an E. coli bacteria. The colors, of course, were the choice of the person who rendered it. I have no idea what color those things are in the actual world. So I'll be talking specifically about 3 results. One is infection prevention. That is, I'll talk to you about what got me into this game, making an E. coli impervious to attack from virus. The second thing is a couple of talks that just came out in January of this year, reports that came out in January of this year, living longer and better, trying to extend the lifetime of, in particular, eukaryotes. And creating, and the last will be just a thing that came out in March, creating egg cells from males. So the first is the triumph over viral infection. So
that figure on the right, which was on the cover, are you still hearing?
No.
Okay. Shows the virus attacking an E. coli virus. The virus is shown is the bacteriophage T7NCL. I don't know why you would want to know that, but there it is. This conflict in the micro world's been going on. It goes on all the time, and probably has for billions of years. Viruses are collections of DNA and RNA and some protein that are about 1/100th the size of a bacteria. Viruses, as you recall, are not considered to be alive as they're unable to reproduce their own. Viruses must reproduce by injecting their viral RNA and produce the proteins and DNA and RNA that are reassembled back into new viruses. A table on the right kind of shows some relevant quantities, understanding the scale of this individual battle. The scale of the warfare is astounding. In a teaspoon of seawater, there are about 1 million bacteria and 10 million viruses. Viruses kill about 20 to 30% of the ocean bacteria every day. But somehow that ecosystem remains in equilibrium over this long period, where the bacteria keep evolving to deal with the viruses that come along, and the viruses keep changing to avoid the defense mechanisms
that the bacteria are putting together. I'm not quite understanding what's going on there, but
in causing the bacteria to create the viruses many times over, the virus eventually can kill the cell and end up releasing over 100 copies of itself. The virus...
I was going to say, okay, I'll just shout. So the figure on the right shows the sequence of outcomes. In A, before the virus attacks, B, the virus attacks and inserts its its DNA into the body of the E. coli. The E. coli has some things in it that have a memory of things that have previously attacked it, and they're CRISPR-like critters, and they can go out and chop up the DNA and RNA of the virus, and if they win, you just go through that cycle again. If not, the virus ends up making a copy, many copies of itself, and ends up killing the cell. I just wanted to show you that that picture or image of the virus is actually the way they look. It's not an artist's conception. This is a view of that virus taken with an electron transmission microscope. So it's a real honest-to-God photo. It certainly looks just like it is. The virus is a really simple critter, just an arrangement of proteins. It's DNA shown here, which when it lands on the bacteria, it inserts that DNA into the body of the bacteria. And we've been talking about E. coli as the particular bacteria. So it's a pretty simple stuff. Now, the bacteria build a response to viral attack via evolution, just as the phages do. Recall in the fourth seminar when we showed how an E. coli produces the proteins it needs. It first copies the gene for that protein off its DNA onto a strand of RNA. A ribosome in the cell then attaches to the head of the mRNA strand and sequentially matches the nucleotides in the mRNA strand with the 4 anti-nucleotides on that tRNA fragment.
Which then?
It's happened again. Which carries the amino acid corresponding to its 3 anticodons. So the codons on this piece of tRNA are down here, telling you they're actually, in this case, anticodons that will match with the messenger RNA, and associated with each one of these codons is the proper amino acid that the ribosome ends up reconstructing into a chain of proteins. Those proteins are the thing that the virus needs to rebuild itself and continue its existence. Now,
this very imaginative person, George Church, had the idea that bacteria could be made impervious to viral attack by using the redundancy that occurs in the cell's translation of codons. You recall, the codon is a sequence of 3 nucleotides. For example, cytosine, adenine, that specify for a particular amino acid, proline in that case. So you see over here, CCA, there it is, and it tells you get proline. So because there is a sequence of 3, there are 4 times 4 times 4, 64 possibilities that you can get in any sequence of 3 of the 4 nucleotides. But there are only 20 amino acids that you need, which allows for a great deal of redundancy, as you see in the Codon table over here. For example, here's lutetium with 4 codons here, and I think there might be another one somewhere. So yeah, there's 2 more up there. So there's 6 codons that could prescribed for that. And you presumably don't need them all if all you really need is that particular amino acid. So the Church's plan was to synthetically construct the DNA of an E. coli using the redundancy in the codons so that certain of these tRNAs would not be produced. He chose the synthetic construction over CRISPR editing to assemble the DNA because, as one of his assistants observed, it's not like you're editing a short story. If you're changing so much of it, you might as well just rewrite the whole damn thing. So that's what they did. They just went out and synthetically wrote the whole 4 million base pair piece of DNA, and then had to insert it into the DNA of a living thing piece by piece. But at least it was in the form that they wanted it. So the E. coli, so you try to raise an E. coli colony that would not be dependent on those missing codons. The genes that code for producing the tRNA that transcribe for the amino acid valine using these codons, GUU. There's valine. There they are. There's a couple of them right there. Would be removed from the genome, and the E. coli would not be dependent on those missing codons. Whereas when the virus injected its DNA into the E. coli, it would need that particular tRNA to construct the proteins that it needed to make up itself. And by 2018, when I think I gave a talk with Lester, it appeared that the Harvard Med School group, which is where Church works, was on the verge of success. They had an E. coli colony that they called EC Syn for synthetic, and 61. It only used 61 codons rather than the whole 64. Alas, it did not work. They went out and just started taking various stuff from sewer water and various other bad spots and seeing if the E. coli was impervious to them. And it turned out that there were some viruses out there that had no problem getting around their thing, and they found out it was because those viruses carried their own tRNA. So what are you to do? Well, we had given this talk in 2018, and I was waiting for 5 years for some result, to read something about it, until lo and behold, it turned out that it didn't work. We didn't know that. They didn't admit to the fact that it didn't work because the publication just came out in March of this year. Mere elimination of the tRNA wasn't sufficient. You had to resort to a well-known human trick, deception. So what you do is in 2 of the 6 ways for specifying for the amino acid serine appears on tRNAs with the anticodons UCG and UCA. The sequence on the E. coli genome for expressing the tRNAs that correspond to those codons actually had serine removed. Had serine removed from them, and they actually ended up in the place of serine putting in leucine. Where is this? Here it is. Rather than that amino acid serine, that those tRNA carried the amino acid leucine. The plasmid would be carried forward in all these particular E. coli. They now changed the name to SYN61SL. That's for serine and leucine. The leucine has very different properties than serine, so when the peptide chain formed with the substitution of leucine in place of serine, it would fold into a mesh rather than the proper protein, thus foiling the virus because it couldn't reassemble in any way with that messed up protein. So here you had a piece of transfer RNA that you made not in the genome any longer, but in a plasmid that you inserted into the E. coli. And that made two things, a thing that had the anti-codon for leucine, actually providing serine, and in the other case, just the opposite, providing leucine where serine was indicated. And that was apparently sufficient, as I show you in this diagram here. Here's the bacteria squirting it in, making a chain of stuff. When it got to the transferred RNA, trying to read serine and leucine, it got the opposites, and it made a bum protein, which ended up being the end of the virus. So we believe we have developed the first technology to design an organism that can't be infected by any known virus, said one of the research guys working on the thing. We can't say it's fully virus resistant, but so far, based on extensive lab experiments and computational analysis, we haven't found a virus that can break it. So this plasmid is a little circular, double-stranded piece of DNA that ends up making these unusual critters that the E. coli itself doesn't depend on, but ends up screwing up very nicely any virus that ends up attacking. So,
May I ask a question here?
Sure, please.
I don't quite get why the bacterium itself. Oh,
I do not understand why the bacterium itself is not upset by that. Because some of their no doubt, many of the proteins would contain those very same amino acid codons.
No, they have raised that colony to be independent of using that particular codon transcription for leucine and serine. So you raised a colony that has been that doesn't make use of those particular codons. So you don't have a genome that produces anything that goes out and looks for that particular set of codons. Because remember, the tRNA is manufactured by the gene, and if you're not carrying it in the
let me
gene.
think a little longer about that. I don't see it right off. But
Okay.
I'll think about it.
Okay.
Is there any secret for putting this in a plasmid instead of the nucleus? Any secret putting this into a plasmid rather than the nucleus of the E. coli? Or the major?
An E. coli doesn't have a nucleus. It
Okay.
just has a plasmid.
Well, wait a minute, plasmid is separate from its own DNA, isn't it?
Oh, you bet. So there's its own DNA which is in there, and then the plasmid, is just inserted into what is this stuff called? Just the liquid in that makes up the cell or the rest of the.
Okay, well, it sounds like there's some secret putting it in the plasmid rather than the main DNA of the cell.
No, apparently it occurs naturally in some cases, and this just happened to be a case where it was useful to put in this particular plasma with these deceptive that produces these deceptive tRNAs.
Well, I think my point is there seems to be a distinction between what's in the plasmid and what's in the cell's own DNA, regular DNA.
Yeah, the plasmid is separate from it.
And that's where they put the goodies.
That's where they put the production of these two nasty tRNAs. Now, you may not like it, but I believe it's true.
I don't like a lot of stuff that's true.
Okay. So anyway, there's one battle won. Now, let's deal with aging. Well, this is one of the most potentially transformative, lucrative, and controversial topics, I think, in biology, is the whole issue of aging. It has all kinds of serious consequences. Remember from previous lectures that mammals such as ourselves begin life, as a single cell, a fertilized egg. We then proceed to an embryo, and so on into adulthood, with stem cells becoming expressed as specific cell types as we mature. The prevalent view during the early 20th century was that mature cells were permanently locked into their differentiated state and could not return to fully immature pluripotent stem cells status. It was believed that cell differentiation was a unidirectional process. When a cell differentiated, it would no longer be able to go back to reverting to being a stem cell. That was all changed by the work of Shinya Yamanaka in 2006. He and his team generated pluripotent stem cells indicated as IPS from adult mouse fibroblasts. Fibroblasts are just normal, simple cells that serve, for the most part, as connecting stuff. When you have a wound, they play a good role in helping it to heal. IPS cells very closely resemble embryonic stem cells. Such cells are the in vitro equivalent of the part of the cytoblast, the embryo, a few days after fertilization, which proceeds to become the embryo proper. Yamanaka's group could show that their IPS cells were pluripotent, that is, capable of generating all cell lineages of the body. Later, he and his team generated human IPS cells from human adult fibroblasts. Again, they were the first group to do so. So a key difference from the previous attempts by the field was his team's use of multiple transcription factors. They started with 24 transcription factors known to be important in the early embryo stage. But in the end, they were able to reduce that number to just 4 transcription factors. Again, I don't think you need to remember this, but there they are. SOC2, OCT4, CLUF4, and those 4 cells prescribe proteins that are highly expressed in similar levels in IPS cells and in stem cells, embryonic stem cells, and the expression of all 4 of these transcription factors decreases when the cell undergoes differentiation. I have that comment that you may have noticed that there was a lot of publicity at that time about that discovery, because doing any kind of research with embryonic stem cells was viewed as a serious no-no by many, many people. The fact that you now could get them without getting involved in fooling around with embryos was a great advance. I think now we're beginning to see the impact of Yamanaka's work on cells extended now to their effect on the whole creature. There's a few reminders that are necessary, and this is what we saw last week. Remember, each and every cell contains a copy of the entire cell's organism. The organism is made up of various kinds of cells, so some epigenomic, that is something external to the genome itself, must come into play to specify what piece of the genome is expressed in, say, in a particular cell, say a muscle cell or, say, a liver cell. So while the genome remains the same in every cell, its expression must be controlled by factors external to that to the genome. The very large size of mammalian genomes, remember ours has 3.6 billion base pairs, employs histones to wind up and compact the genome so it can fit into a cell. So here's a histone. It's made up actually of 8 pieces of protein, 2 of which are the same, so there are 4 different proteins arranged in a particular way. And the DNA comes and winds itself around it so that you are able to sort of neatly package the DNA first into these histones, and then the histones end up looking like this, and then they get repackaged even more tightly into a form like this. Remember that the human genome, if you unwrapped it, would end up being about 6.5 feet long. The histone binds these things up in such a way that you actually can fit that genome into every one of the roughly 30 trillion cells in your body. Now, packaging several histones together makes it difficult to access the DNA tied up in that pack. And notice that there's some methylation shown attached to each of the histones. And it turns out, as we mentioned last week, that methylization of the nucleotide, in particular cytosine, in the cell's DNA, is an important epigenomic factor. It expresses that it represses the expression of a genome when it occurs in the gene's promoter or enhancer. And if you recall, the methylization of cytosine is just simply the addition in this 5 spot, 1, 2, 3, 4, 5. So that's why it's often called 5MC, the 5th position methylization of cytosine. And it can either repress or in some cases enhance transcriptional activity. And these epigenomic factors are going to be exploited in the following research. So 2 articles dealing with the aging of mice appeared this January, one in the journal Cell and the other on the Cold Spring Harbor preprint server. The articles represent what I think are major contributions to the field. One reported that not only can mice be aged on an accelerated timeline. But the effects of that aging can be reversed, and some of the biological signs of youthfulness could be restored to those prematurely aged mice. The notion of restoring youth is captured in the term rejuvenation. That reversibility makes a strong case for the fact that the main driver of aging isn't mutation to the DNA, but rather miscues in the epigenomic instructions for cell repair that somehow go awry with age. The rebooting of the aging comes about in the form of gene therapy involving genes that instruct the cells to reprogram themselves. In the case of mice, the instructions guided the cell to restart their epigenomic configurations that define their initial identity, for example, as kidney or skin cells. Those are two kinds of cells that are particularly prone to the effects of aging. These genes create the proteins in the suite of the so-called Yamanaka things we talked about, stem cell factors. A set of 4 genes that the Nobel scientist Shinya Namasaki discovered that can turn back the clock on adult cells to their embryonic stem cell state so they can start their development or differentiation process all over again. Now, it's important to note that neither of the groups I'll talk about use the full set of the 4 Yamanaka factors. They use only 3, SOX2, OCT4, and CLUF. There's no vowel there. The 4th one was not used. After all, you don't want to go all the way back to stem cells. Think of a cell in your brain or your heart reverting to stem cell status. Part way back is quite good enough. You might want to start back to your 50s, not 5 days after your fertilization. So you see this kind of stuff can be a bit tricky. Apart from their use of the 3 Yamanaka factors, the research approach of the 2 groups was rather different. While the research explained in detail in their respective publications, they're really far too technical and full of acronyms for me to directly transmit the content to you. I'll do the best I can. The Cold Spring Harbor preprint was authored by a group from Rejuvenation Bio. That's a private company that was co-founded by George Church, the chap I mentioned earlier. That group worked with very elderly wild mice, comparable in age to something like somebody 77 years old. And the Cell article was from a large international research group headed by David Sinclair, again of the Harvard Medical School. Interesting how that stuff is ended up what attracted me. So let me start with the article in Cell. It was really annoying. It cost me $44 to look at it for 24 hours with no possibility of copying anything out of it. I have to say the article is very complete. It was 50 pages, so I couldn't digest the whole thing. The senior author, David Sinclair, has a theory of aging that the article provides some support for. He has long proposed that aging is the result of losing critical instructions that the cell needs to continue effective functioning. He terms it the information theory of aging. He says, underlying agent is information that is lost in cell, not just the accumulation of damage. That's a paradigm shift in how to think about aging. Following Sinclair's point of view, the first step in the reported research from that group was to accelerate the aging of selected mice by chemically inducing many double-strand breaks in their DNA. This was done in a manner, at least according to them, as to minify damage to the mouse genome. So here you're making a double-strand break in the DNA, and you're claiming that it doesn't do anything. In a young cell, that double-strand break will be repaired and repaired perfectly. But what they did is feed into a particular set of these mice many breaks. These breaks in the selected mice went on for about 3 weeks when the mice were about 4 to 6 months old. There were no immediate ill effects or mutations that you could see in those mice, but after about 6 months, those ice mice, ice stands for irreducible change to the epigenome. These mice showed definite signs of aging compared to the control group. So here I show you an ice mouse, and here's his brother or sister, I don't know, from the same litter. This is now 6 months after he's had this dose of a lot of double cell breaking going on. So the mice didn't just look older, but also showed biological aging in the loss of methyl groups at many sites in their genome. And I would say this was in line with Sincare's view that aging results from epigenomic changes to the genome expression that result from maybe faulty folding of the genome or changes to the methylation status of some of the genes. The genome itself is unaltered, but its expression is altered because of the multiple breaks and repairs in a way that appears identical to the signs that show up due to aging. In the next phase of this experiment, the aging of the ice mice was reversed. They successfully reset that epigenomic changes in the ice mice using 3 of 4 of the Yamanaka factors, and it resulted in a 57% reversal of the symptoms of biological aging. I lifted that 57% out of the article, but was unable to give you any of the evidence for the number. An adeno-associated virus, AAV, was used to deliver the OSK, the Yamanaka factors, into the genome of the mice. Using the AA virus has become a preferred platform for doing this kind of gene delivery. The principal reason appears to be that the virus itself has no pathogenicity, and it can also affect it can infect non-dividing cells and has the ability to stably integrate into the host genome at a specific site. It appears that somehow mammals retain a back copy of the youthful epigenomic information that you safely can get access to by using those 3 of those factors to restore the function of old tissues. Maybe it's akin to something like rebooting your computer. I don't know. The article on the preprint server was free and allowed copying figures. The approach of the Rejuvenation biogroup was far more straightforward. They just started with a bunch of old mice and investigated the possibility of extending their lives. They also used the same adeno-associated viruses to deliver those factors into the genome of mice that were 124 weeks old. That's and that, as I said, I think that age is something like 77, and the median lifespan of a wild mouse is approximately 129 weeks, equivalent to about 80 years in humans. So the figures below were taken from their preprint. So figure A just shows the procedure in basically using the AAV virus to inject stuff into the mice. In one case, they put in the 3 factors from the Yamanaka suite, and in the other case, sort of a more or less harmless dummy. Over here is the results of what they found. So in the cases where they injected the 3, and these numbers here refer to the number. So there were 20 mice involved in each thing. 20 mice had the 3 factors injected into it, and there were 20 mice that were did not have that. And they also show just a historical thing from the Jackson lab of just wild, untreated mice. And this is the lifespan, and what they show is the percentage of survival after a given number of weeks. So it's quite clear that using those 3 Yamanaka factors considerably extended the lifetime of those mice. This is the lifespan of the control group, and it follows rather closely, especially on the average, the historical wildlife thing. So it does appear that there is some extension of life going on. So here's some more figures. This is the remaining lifespan after injection. So after at 124 weeks. So the longest lived of the injected mice seemed to be something just over 40 weeks of additional life. Notice that in the control group, almost every one except one had perished by the end of 20 weeks. And here it looks like the order of half might have survived longer than that 20 weeks, and in a couple of cases, appreciably longer. Not only did they survive longer, but an index that they use as a frailty index was used here to show the lower the frailty index is, the better. So the control group here was considerably frailer than the group that had the 3 prescriptions. And here again is just a long graph of the history of those 20 mice. And here is the treated group, and here is the extended group. It almost looks like they, nearly reached a factor of 2 extension of the life of these mice. So if you transfer it to 77 to human terms, if you're 77 years old and you start taking this stuff, you're expected to be done in by 80, but there's a good chance that you can live to be 83, and you presumably will feel better. So, and so there's every indication to me that introducing this stuff into the genome of elderly life does have the effect of reducing many of the symptoms and ill effects of old age. And further research is underway to see if that result will stand up in primates. To my mind, it's really too early to jump to any optimistic conclusions. These two experiments are not very detailed in the cause of what has actually caused their interesting result. Why does the OSK cause the restoration of apparent youth? Is it just the resetting of epigenomic factors that explain it, and it's somehow a particular kind of damage that's done to epigenomic factors get straightened out, similar to rebooting your computer? Or maybe a better example is cleaning an old CD, getting your fingerprints off it. To me, this business strikes me as very risky. Yamanaka factors certainly push back cells to their previous stages of their development, but I don't fail to see where there's any real control as to how far. Other researchers were also much concerned with a thing called teratoma. Those are tumors that are made up from a really ugly kind of stuff that's made up from various kinds of tissue, all appearing in a single tumor. Doing this kind of research in vitro seems fine to me, but carrying it out in vivo seems to me to be a pretty risky business. So, I'm sure many of you have heard of this because it got so much attention in the press. In March of this year, a stem cell biology at Kyushu University in Japan reported that their group had created mice with two biological male parents by manipulating the chromosomes inside a stem cell. So in 2016, this group had managed to drive a female stem cell back into an egg cell. Previously, all egg cell creation began with stem cells from female animals. Hayashi's team first took the cells from male mice and turned them into pluripotent stem cells. But they're still male cells, remember. Those are special kinds of cells that can be turned into many other kinds of cells, including eggs. Now, they grew these male cells, XY cells, in petri dishes until some of them ended up losing their Y chromosome. Now, the cells had one copy of the X chromosome, but no Y. So the eggs in mammals are always female, and you need to have two X chromosomes. The team then used what was, I think, the important step in their research, a genetic trick that allowed some of these single X cells to pick up an extra X chromosome or genetically turn them into female egg cells. They then embedded the cells in an artificial ovary that was also produced from stem cells. In the ovary, the manipulated cells developed into an egg cell or acolyte, which then could be fertilized with sperm from a surrogate mouse. This resulted in 7 living pups. So, excuse me, it's important to realize they transplanted 630 embryos formed from such egg cells into surrogate mice. This resulted in 7 living pups that grew normally and were fertile as adults. So the research was reported as these mice had aged to the point that they could themselves have offspring. The low success rate, that is just 7 out of 630, which is the order of about 1%, he says, isn't due to the process of the sex chromosome conversion, but the reality that cells cultured in the lab are typically inferior to those cultured in a living animal. And just the typical success rate that I found in reading it varies all over the map, but it's as a round number, 50%. So that 1% number is really quite troubling. Of course, the researchers haven't successfully produced human eggs and sperm cells from stem cells, but this particular student who is now at the University of Pennsylvania, who worked with Hayashi, thought that in humans we're way behind, that he thinks that producing human gametes might be technically feasible within a decade. That's a kind of concerning thing. So, thank you all for listening. I've learned a great deal in preparing this series, and in particular the time that I had to invest in learning a bit about cell function allowed me to appreciate, first of all, the reports on current research, and it was also rewarding to realize how wonderful, surprising, and unexpected life is. And as a physicist, I can say, a physicist can tell you what's possible, but a biologist tells you what is. Thank you.
I don't know if I can hit. The first time I remember hearing about it, bacteriophage, was probably from I used to read books about doctors all the time. It was a book written in the late 1940s. It was Aerosmith. The first time I read about bacteriophages was in a book by, I forgot whose name is called Aerosmith. He was talking about bacteriophages to attack bacteria and so forth. He never did find quite the cure. It came close to it.
This isn't simply a cure, right? This is an ability to render a whole group impervious to further attack.
I think a lot of us here would really enjoy something that would extend the quality of life. But if you look at the world population, we got way too many people already. And I'm not really excited about all of us doubling our lifespans.
Well said.
I had a hard time measuring whether those mice with extended lifetimes were happy.
Hi.
Thank you very much, Gerry. I wanted to ask about the mice in panel E, and that relates to the question that was asked in that experiment in the cell paper. First of all, did the group, with Sinclair's group, I assume, check on the mental acuity?
I'm having trouble hearing. But
Just closer to your mouth.
Okay. Can you hear me now, Gerry?
Better.
Good. Did they look at the mice with extended life that they had altered the epigenetic pattern of? Did they look at their cognition?
At their condition.
Cognition, their problem solving, maybe to answer Al's question, if they get not just get younger but more greater mental acuity.
Yeah, I have to say the article was 50 pages long, and by the time I got to what the results were, I was failing. So I really can't answer your question about what the final stages of that were. It was clear that they were far more sophisticated than the spin-off from Church's group. They just kept track of whether they lived or died for the most part. So I can't answer that as well as I should be able to.
Yeah, we can check on that, but I think that would be to the point that Al was raising. If we get older, can we also rejuvenate mentally?
Yeah.
And mice can help us with that experiment.
I'm pretty sure there were no maze problems or anything like that mice were subjected to later on. But, boy, sorry, I don't have a better answer.
Gerry, in the administration, there's 3 factors
Yes.
to extend life. How are they administered?
They were administered using a virus that they were able to inject the genomic material into the virus's DNA, and the virus then pushed its DNA into the cell, and it was the kind of thing that it got incorporated into the genome of the mouse.
And
This
then-
is
When you talk, I thought the aged mice were administered these 3 factors to.
Yes, and those three factors were added to the DNA of the virus.
Yeah, and the virus then infected
The
with
virus then infects the mouse,
the elderly mice.
the elderly mouse, and
Well, I don't see why the older mouse, why the target there is a gene, is the I'm sorry, the egg or sperm cells. I mean, it's.
yeah. No,
I'm wondering where those
okay.
factors ended up in the.
So I think they were, to my mind, rather weak on that. In earlier research that the Sinclair group had done, they cured glaucoma in aging mice by inserting these factors into the really right near the eye.
Right.
And so there was a kind of localization. I had a hard time just understanding how the three factors would end up in just the right places that you wanted it to be.
Yeah,
I have.
but you're not clear, or don't you recall how they administered them? Did they just inject them into?
Well, in that photo or the image I showed, the group reporting in the Cold Spring Harbor thing looked like they were injecting it into the eye of the mouse.
That's for the glaucoma stuff.
No, that wasn't for glaucoma. That was unless they just lifted the figure. Yeah, for some reason they just show in the figure, as in the article that they wrote, they showed the injection being put towards the mouse's eye.
These also have inducible promoters, as far as I can tell from the figure, to turn those specific genes on. That's how I read this figure.
But it,
Yeah, factor jobs, genes.
but would it go to every cell in the body?
Not necessarily. Well, the inducer would.
Yeah, the inducer would.
Where the viruses go, I don't know. I share Lester's concern about what cells are getting the virus.
Yeah, well, to me, that's the whole concern about that approach. They seem to be very indefinite about how this miraculous reduction of aging actually comes about. How does the stuff go to the right cells?
And maybe it goes into blood cells, and this blood is what's circulating the Yamanaka factors after synthesis. I don't know, but it's a fascinating approach and paper. Thanks for sharing that.
There's very little mention of how the spread of those factors go about in the articles, and that was one of the things that upset me about doing it to living creatures. It's fine to do it in a test tube.
Well, let's get a copy of the paper and then we can, those of us interested can read it.
I was not allowed
Yeah.
to enter that sacred world. Beth?
It sort of sounds like they threw stuff against the wall to see what stuck.
Well, yeah, I'm not sure. It is interesting that both papers came out the same month. One had the feeling there was competition going on, and oftentimes competition will lead to papers put out before they're thoroughly digested.
And they're both from Boston, so they've probably heard about this.
Oh, they certainly did.
Yeah.
They're both Harvard Med School people. And neither of them seem very shy. Okay, anything? Henry? No. Okay.
I don't know enough to ask a question.
Class dismissed.